morning. It's nice to be back in California, I gotta say. Thanks, Jim. Jim just really wants to hear what I have to say.
That's right.
So I'm in North Carolina right now, and so I spent a lot of time thinking about what I can do to get back to California. And this talk kind of developed out of that sort of drive. I really enjoy working in California, Flora, both as a botanist, probably like many of you, going out learning the flora, and then also some sort of from more of an academic perspective, looking at the evolution of the California flora. And just like with the vascular plants, the mosses are super diverse in California, and that makes it not only fun to sort of go out and learn the flora, but it also makes California a really exciting place to study the evolution of the moss flora. And so I'm going to talk a little bit about that today, with the emphasis on molecular data and what that is contributing so far and what it can contribute in the future to our knowledge of the Bryophyte flora. And I'm going to be talking mostly about mosses today, but there'll be a few slides that include the liverworts. And that's mostly just because the Bryophyte flora of North America has about 4 times as many mosses as liverworts, and so they definitely have the driving force. So what I'm going to talk about today is what is the Bryophyte flora of California, kind of in the context of the Bryophyte flora of North America, because it's very relevant. Like as Lena was talking about, a lot of times Bryophytes have really large ranges spanning several continents, and so you kind of have to zoom out and look at the whole continent to understand what the California Bryophyte flora is. And then also, how well do we know it? How good of a job are we doing right now documenting the flora that we do have? And then I'm going to talk just briefly about the impact that molecular data are having on that, that kind of search to understand the flora, and seeing whether or not they're actually having a big impact. So we know, for example, in the vascular flora, that molecular data are having a huge impact. And so all of you that deal with name changes and the Jepson manual and things like that, that molecular data are really critical in understanding evolutionary relationships. And then, just as kind of a finishing note, I want to talk about what molecular data are telling us about the origin of the flora. And I think, for me, that's what's one of the really interesting things. And the answer, I'll tell you now, is not much, just because we haven't looked. And what we do know tells us that it's really complicated and interesting in some ways that the vascular flora is not. So that's really cool, and I'll talk about that. So the first thing that you need to know about bryophytes is that endemism at even the continental scale is quite low. And so for example, these data, and I have a couple slides of data from the Flora of North America project. So the Flora of North America has 3 volumes on bryophytes, 2 on mosses and 1 on liverworts and hornworts. And the 2 moss ones are now complete. So this is actually a really nice time to be understanding the kind of continent level patterns, because we have, for the first time, kind of all of these data in the same place. Of course, given the fact that things change over time. But so there are a little over 1,400 native North American mosses, and about 21% are endemic to the continent. So that's pretty amazing if you're coming from a vascular plant background. 4 out of 5 species you encounter also live on another continent. And so when you think about biogeography, floristic patterns, it's something that they have to keep in mind. So if you're talking about the California flora, what does that actually mean, given the fact that most of these species also live elsewhere? And endemism in the California floristic province is even lower. Actually, I can't remember if the vascular flora, that number might be totally wrong. Anybody out there, what's endemism? Is that about right? Okay. Close enough, all right. So anyway, it's, the number for the mosses is right. So it's about 5% for them. So much, much lower than for North America as a whole. And just to give you a real general sense for what bryophyte diversity looks like in California, if you're not familiar with it, these numbers are the number of species as of a few years ago And this is just mosses, not liverworts, in the different ecoregions of California. So you can just get a general sense that a lot of the diversity in the Northwest and the Sierras, and then as you go south from the Northwest to the Central West to the Southwest, it drops off. And then, as you can imagine, it drops off towards the deserts also. And this figure was, like, putting together this was what really made me wonder what I'm doing on the East Coast. And it's really exciting. So on the x-axis here we have time. So moss nomenclature officially starts in 1801, and then so we're going to go all the way to the present. And then on the y-axis we have the accumulated number of endemic species in North America. And so the reason that I'm looking at only endemic species is because most, the species that occur, for example, in North America and also Europe, often they were described in Europe. And so this is the date of first publication for all our endemic North American species. And so if you can imagine if you see a line that looks, a lot of coffee this morning, if it kind of bends over like that, you know that we're doing a really very good job. And we have a good sense of the diversity. You get a line that sort of goes straight up, or a line that kind of curves up like that, then we have a lot more work to do in documenting flora. You ready? Here it is, all right? So this is really exciting for, for people, I guess, that are really interested in documenting diversity. That line is not tapering over, right? It's especially look at the last 10, 15 years, it's just shooting up, which is really, really cool. And that means that people are out there looking for mosses, and they're discovering new mosses. And so you can see that this is not just a straight slope, like there's some humps and bumps in there. And a lot of that actually is just explained by if you take the Western North American mosses and the Eastern North American mosses apart. And so you can see the Eastern North American mosses just kind of gradually curve up. And that one, it's definitely not a plateau, but it's definitely kind of, it's drifting off. But in Western North America, you see like between 1850 and 1900, there's this really steep part, which obviously coincides with like the railroad surveys and the big kind of exploration of Western North America. And then what's really cool is right at the end here, so all of this kind of like shooting up at the end, a lot of that is from Western North America. Yeah, Jim. What's even more interesting is if you look at species that are endemic to California, and again, this is a really low number, it's around 30 species. But a lot of what's happening in Western North America in the last 10 or 15 years is a direct result of things that have happened here in California. And so I do a lot of molecular phylogenetic work, and so I was like, oh, this is cool, like we're, things have been good, but then once molecular data come along, we're really just pushing the boundaries and getting to know the flora. And that's not completely true, and I'll show you that in a second. So just briefly to summarize that first part, endemism is really low in California and in North America in general, but our flora is really distinctive. We do have endemic species, and we have a lot of West Coast endemics that are primarily Californian species. And then really importantly, a lot of the recent work shows that we have a lot to do. That curve is not even close to tipping over. So this is a really exciting time to get involved in bryology. So the second point in my three-point outline, so out of that discovery, what's the impact of molecular data? So what I want to do is I'm just going to kind of zoom in on this little hump right here and show you what species discovery in California looks like. So this is the same set of axes. So we have time on the x-axis from 2000 to 2014 and the accumulated number of species. But here I've separated out the species that we've discovered through kind of what I call traditional methods. So going out into the field, spend time in herbarium, versus in red, that's the added influence of molecular data. And as a molecular kind of biologist, I really get excited about, so here in the last 5 years, we're just kind of ratcheting up and up and up. But the other really kind of cool story is like, look at how many species we've discovered, given that we have, maybe a dozen people out there really kind of actively looking and, sending material out to specialists and stuff. There's a ton of stuff out there to find. This curve is nowhere near flattening out. It's just going up and up. So it's just really, really an exciting time to be doing both molecular work and also just to be getting out and documenting the flora through kind of tried and true methods. So are they making, molecular data making an impact? Yeah, they are. But the real kind of story here is that over the last sort of 20 years, and a lot of that influence is from Brent Mischler and Jim Shevok and Dan Norris, kind of getting people excited and bring people together through the forays and Jepson workshops and other things. A lot of the big gains we've made have been just through the good old-fashioned getting out and looking at plants. So then what about the origin of flora? So we know, for example, that think about things like Arctostaphylos, the manzanitas, where you have 50 or 60 species that live just in California, and within that genus, there's only a couple of species that live outside of California. So you have this like massive radiation, all this diversification, new species all over the place in California. And it's a really good way to study speciation because you know that all of that stuff happened right here in California. You can kind of understand the evolutionary process by looking at these species. But if we only have 5% endemism in California, we can't really do that with the mosses, right? Because you can't, if you have 5 or 10 species in a genus and they all live in California, you don't necessarily know that they evolved here. Maybe they evolved somewhere else and came over, and we don't know who the closest relatives are. And so that's kind of what I want to talk about, and this just sort of summarizes that. But the big question that I'm kind of interested in right now is, what can we say right now about where our endemic flora came from? And is it the same story that we see with the vascular plants? Is it a different story? What's going on? And so I surveyed the literature, and there's really only like a half a dozen papers that have enough kind of sampling of California species that you can actually say something. So again, it's a really exciting opportunity for people to get involved. It's more limited because you have to have a molecular lab, obviously, but as we kind of encourage young people to get involved in bryology, those people can go and do graduate studies and actually undertake some of these projects. So I'm just going to focus really quickly on 2 kind of really nice case studies of looking at diversification of mosses in California to give you a sense for what the general patterns are. One of these is Homalthesium, which you guys have met before if you're in here for the last couple talks. It's got about 10 species worldwide. 7 of those are in Western North America, and it's got 3 species that occur in Europe and Eastern North America and Asia. So again, we have this genus with stuff kind of all over the place, but centered in California and Western North America. And so a recent molecular study by Houghton and colleagues, a European group, generated a molecular phylogeny of this genus. And the names and stuff on this phylogeny are not important, but the colors represent geography. So the black dots, like all these things up here, that represents North American, Western North American species. And then the white and light gray are the other ones, so Eastern North America, Asia, Europe. And so what we see here is, first of all, that probably this group evolved in Western North America. So again, these are the species at the tips, and like this is the ancestor of that species and that species. And as you go deeper in the tree, that's going further back in time. So the root here is in North America. And what's cool is that we have 2 nodes. So this one leads to 2 species, and then this one leads to 1, 2, 3, 4 species. And both of those have an ancestor reconstructed in North America. And they actually did a dating study, and so they estimated that both of those divergent states were within the last 5 million years. So this is a case where we see something that is actually not too dissimilar from what you would maybe see in like a wind-dispersed vascular plant, maybe. So you get a diversification of things outside of Western North America, and then you get a bunch of diversification within Western North America. So here we have species actually evolving in place, more or less. And we see a little bit of a different case in the genus Orthotrachum. So this is another one that we've talked about today already. It's a much bigger genus, has about 135 species worldwide, with 55 in North America. And 29 of those are endemic. And so this is the most important genus for endemics in California. And so this has been studied quite thoroughly by some Spaniards, led by Rafa Medina, who just published a PhD dissertation on this group. And one of the papers that he's recently published is looking at this species, which is called the Orthotrachum tenellum group. And formerly it was thought to be one species that occurs in California and the Mediterranean region of Europe. But his work has showed that it's actually 6 distinct species. And even further, those 6 distinct species are not each other's closest relatives. So you get a whole bunch of kind of convergent morphology in there. And what this looks like on a phylogeny, again, don't worry about the details, but the colors here are represent geographic ranges again. And so dark blue is California, light blue is Western North America, including California, and red is the Mediterranean, and then there's one Chilean species right here. And so what he found was that there, within this clade of 19 species, at least 3 instances are situations like, just look at this top one up here, where you have a Mediterranean species sister to a Californian species. And so in this case, it's very clear that one species probably already existed, and then you have a long distance dispersal event to another continent, and now you have 2 sister species on different continents. So within this little clade of only 20 species, you have at least 3 really nice cases of that. And then on the other hand, you also have at least 4 cases where we have sort of more local divergence. And by local, I mean sort of just within Western North America, sort of more typical of what you would see with the vascular plant. So in this case, you have a Western North American species sister to another Western North American species. So within Orthotrachum, you have both cases, and you have things that are diversifying sort of at this global scale, and you also have diversification happening at this more local scale. And I think that this presents sort of some difficulties that you don't really encounter as much in studying vascular plant evolution. So you have to take this more kind of global look to understand which lineages actually are diversifying right here in California. So this just summarizes that you have both of these processes happening, this kind of like global stuff and this more local stuff. And so to understand how our flora is evolving, we're really going to have to take a pretty broad view to make sure that we are really studying species that are closely related to each other. So some just real general conclusions. Species discovery in California is really, really active. And that involves both molecular data and also just good old-fashioned field work and herbarium studies. And molecular studies are beginning to contribute to our understanding both of species discovery and also the origins. But really what we need is just a lot more studies. This is not a simple case. In every case we look at, things become more and more complicated in a very interesting way. So if you have any questions, I'll take them.